Secondly, if anybody needs to take exams through RDS, that is an option. You may know there's some paperwork to fill out, so you can bring that by any time. And my third one was also on RAMCT are some old exams. Actually, the exams that we had from the spring semester. And even though we've divided up the material slightly differently in terms of what chunks go on what exams, the format will be really the same. So if you'd like already to go in and see what's an exam going to look like in this class, you can do that. It's sort of a mixture of different kinds of questions. Multiple choice, true, false, short answer, fill in the blank, label a diagram, different sorts of things, which I think is kind of nice for different types of learners. So hopefully that'll be an advantage for you. The keys to those exams are also up there too, so if you wanted to use that as a study tool, so study like normal and then take the last spring's exam and see how you do, that would not be a bad idea. So it's just some extra resources. All right, so today we're going to start talking about sea urchins, and this is our first model organism, and you're going to see some of the features that led scientists to choose the sea urchin as a species to try to learn something about fertilization. So as we talk about the sea urchin, keep in mind that we're really talking about the process of fertilization. And what is it that this is going to accomplish for the embryo? What sort of special features or mechanisms does the embryo have to employ to make sure that fertilization is successful? So I use this term mechanisms, and that's intentional because we don't want to just learn a bunch of names or labels or facts, but we really want to understand how these processes are happening and why the organism, is doing it this way. So what would be needed for fertilization to be successful in the sea urchin? So just to introduce the sea urchin, what we have here is an adult individual. They're a marine organism, and they live out there, and males will send out sperm that has been caught up by the water and brought over into contact with eggs laid by the female. And one of the reasons that this was chosen as an organism to try to learn something about fertilization is the eggs are very large. So you can collect these eggs in the laboratory, you can collect sperm in the laboratory, you can put the two together and get fertilization events to happen right there in the petri dish. So that's very handy because that means you can manipulate the conditions. You could change things like the number of sperm, the pH of the water. You can explore all the different conditions that would be necessary for really nice fertilization in this organism. Secondly, the eggs are large, so you can have a better chance of actually seeing and observing the important events that are happening during this really early stage. Just feasibly it would be much more difficult if these eggs were 1/100th of the size that they are. In lab, you're going to get a chance to actually do this experiment, so I think it'll be pretty cool, and hopefully you'll see for yourself some of the same events that we talked about today. So let's have a look at the gametes. So that is the sperm and the egg for this particular species. And one thing that you'll notice right away is that these gametes, these single cells, are very specialized for the function that they have to carry out in life. So the sea urchin sperm have these really, really, really long tails, as sperm generally do. And then they have a very, very compact head to them. All of the DNA is up here within this head portion, and it's in a very condensed state, just really to make it smaller and more streamlined. And that's because these sperm have to swim, a relatively long distance to find the egg and then fertilize it. So the sperm come equipped with a long tail that has microtubules within it to give it swimming power. It comes equipped with some mitochondria that are going to provide chemical energy. It comes equipped with a centriole, a nucleus, and then a couple of other key features. A little blob of actin, and we'll tell you what that's all about. And then in front of that, another small membrane-bound pouch or vesicle called the acrosomal vesicle, and we'll talk about that too. So sperm are numerous in number and pretty small. The eggs, on the other hand, they look very different. For one thing, they're large and they're stationary. They have, in the very middle shown in blue, a nucleus containing haploid DNA. So sperm are haploid and nucleus is haploid. When we get them together, we restore the diploid nucleus. They also have mitochondria, as well as other cellular organelles kind of spread around. The gray here indicates the plasma membrane for this single cell. So they have a typical plasma membrane. And right underneath it, all of these yellow circles indicate a number of very small vesicles, or small membrane-bound sacs, that are filled with some proteins and other contents that I'll tell you about. So they're all sitting here right underneath the membrane, waiting for fertilization to occur. Then we have another layer shown in red. So this is just a layer of lipoproteins. It's called the vitelline envelope. And that's going to be important in fertilization. And then around that, all that yellow mass is called the jelly layer. And this is some kind of glutinous material that's around the outside of the egg. So these two gametes look very different, but they're each specialized for their function in life. So one of the challenges for sea urchin, sperm and egg, is they need to find each other. So there are going to be a couple of different mechanisms that sort of help that process to occur. They're physically separate in space, but they need to get together. And one of these is chemotaxis. So this is thought to be a chemical signal released by the eggs that attracts sperm to come toward this source of chemical, or that is toward the egg. And moreover, this is a chemical that is recognized species by species. This actually accomplishes a second goal here, is that you're going to get the correct species of sperm to be drawn toward an egg of the appropriate species. So if you have a different species fertilizing an egg, that's not really going to be very workable for the survival of that embryo. So in this picture, you can also see the size difference between these two gametes. And the egg is huge and it's covered with 100 or more different sperm, and they're all in the process of trying to complete the events of fertilization and be the first and hopefully only sperm that fertilizes that egg. Okay, so what happens here is when the sperm contacts the jelly layer, this sets in motion a series of events that will change the morphology of the sperm. So compounds present in the jelly layer are going to trigger calcium ions to enter into the sperm cell. And when that happens, when calcium enters into the interior of the sperm cell, this vesicle here, known as the acrosomal vesicle, is going to fuse or meld with the plasma membrane. So inside we have this vesicle covered with plasma membrane, and that plasma membrane and the outside cell plasma membrane are going to join up together and become one. And effectively that will release the contents of this acrosomal vesicle to the exterior of the cell. Question?
Can you please repeat that again?
Sure. So once we have, move this chair here. Once we have the sperm come into contact with the jelly layer, we have calcium rushing into the interior of this sperm cell. The acrosomal vesicle, surrounded by plasma membrane, will fuse then with the cell plasma membrane and release the contents of this vesicle to the exterior of the cell. So let me show you the next slide, because that is a picture of this event. Okay, so initially here we have the sperm, that condensed nucleus with the DNA, this blob of actin monomers, and then a membrane-bound sac shown here in green. So initially this is completely surrounded by membrane, but then after the sperm contacts the the jelly layer, now that membrane of the vesicle and the membrane of the sperm cell fuse, and all those green dots indicate the contents of that acrosomal vesicle being exocytosed. Does that make sense? Okay. All right. So now let's look at another event that's happening here. Initially, we have a number of actin monomers. So these are called globular actin because they're basically in more of a individual rounded shape. But actin has the ability to polymerize and become a long filamentous type of molecule. And so that is exactly what tends to happen. So after the acrosomal reaction is occurring, these globular actin are getting together and forming a long actin macromolecule, otherwise known as a microfilament. So you may remember that from cell biology, that we have microfilaments composed of actin and microtubules composed of tubulin. Yes. So these are some of our cytoskeletal components. Good job. Okay, so this, as these microfilaments start to elongate, they are still in the interior of the cell. That is, the membrane of the cell is still surrounding and covering these actin microfilaments. And as they grow longer, they're just going to push that membrane out. And so membrane will be covering the actin microfilaments as they extend. Okay, another thing to point out here is that in the original acrosomal vesicle, on the more interior side, there were some proteins, and these are called bindin. And so as these events unfold, what these bindin proteins, shown here by these little blue circles, these are going to eventually end up on the outside of the sperm. So they're now exposed after the acrosomal vesicle exocytosis, and they're now coating or embedded in the membrane that is on the very tip of that sperm where the microfilaments are. So just some terminology where you have the bindin proteins. This long extension is known as the acrosomal process. And yes, so acrosomal reaction has now occurred. Okay, any questions on this?
So is this process kind of conserved around lots of species, or is this pretty specific just to sea urchins?
So in some senses it's similar, it's conserved to sea urchins or other invertebrates have an acrosomal reaction and an acrosomal vesicle. Later on in the course we'll talk about humans and mammals, and you will see some differences. And as a matter of fact, your book has both in the same chapter, and I used to teach them back to back, but I think it was too confusing because people got mixed up of which one did what. So I'm just presenting sea urchin to start out, and then later we'll come back and look at mammals. So, yeah.
What do the acrosomal enzymes do?
Okay, what do the acrosomal enzymes do? You are thinking ahead, and that's a very good sign, so hold that thought, and we will get to that in a couple of slides. Okay? So this also has the advantage for the sea urchin that these binding molecules can be species specific. So they are going to be binding to the vitelline envelope. So that was a red layer in that egg that we just showed you. So after these acrosomal contents are released, there are enzymes in that mixture that will help the sperm kind of digest its way through the jelly layer. So the sperm gets closer and closer and closer to the vitelline envelope. Finally, when it arrives, these binding molecules are present, and they can bind to receptors present there on the vitelline envelope. So this is another step where species specificity can come into play. So that just aids the process of fertilization for this organism. So these two slides down here, I want to point out this is an immunohistochemistry experiment. So is this telling us something about protein or mRNA? Protein, right. So this is a way that we can ask where is the binding protein be localized. So the experiment here is to to try to ask that question, and they've used an antibody that is specific to this species of sea urchin binding protein. And all of this dark precipitate here, it's not colored because it's a black and white picture, but this is all here right around this acrosomal process, a large deposition of color. In this slice, we have the sperm coming in from the right-hand side here at the acrosomal process, and this is contacting the egg. And all of the binding protein is all located right there at the very tip of that sperm, where we expected it to be, if that makes sense. Okay. So here's kind of a synopsis of where we are right now. The sperm was swimming along happily, was attracted to come over to this egg by a chemoattractant. When it arrived at the jelly layer, the acrosomal reaction was triggered. The contents of the acrosomal vesicle got dispersed onto that jelly layer when the acrosomal reaction happened. The acrosomal process extended. The sperm digested its way through, and now you have binding molecules interacted and bound to the middle of the envelope. At this point, now we have the sperm plasma membrane and the egg plasma membrane are really, really close. And so the first sperm that can make it this far and connect to the middle of the envelope may be the one that actually fertilizes the egg. So when the sperm plasma membrane and the egg plasma membrane fuse together and unite, that essentially is fertilization. So that's going to establish this structure known as the fertilization cone. And now for the first time we have a continuous membrane joining up the sperm cell and the egg cell. The next thing that we'll see happen is actually not only the nucleus, but the entire sperm goes into the interior of the egg cell. So here's an actual picture of what this looks like, because I think the diagrams are instructive, but they're a little misleading at the same time. So these sperm, for one thing, are very, very thin and long. On the surface of the egg, there happen to be these many tiny protrusions known as microvilli, and the acrosomal process, once it gets that close, forms this fertilization cone that looks like this picture shown here. And from that point on, we'll now see more and more of the sperm cells becoming incorporated into the interior of the egg. Okay, so here's a picture of the sperm cell is now diving in, if you will, and actually a transmission electron micrograph. This shows you this really dark condensed material. What do you think it is?
Nucleus?
Chromosomes, DNA, nucleus, yes. So that has been condensed partly for the protection of that DNA, just so that it can stay intact as a single blob. Also, it does not really need to be transcribing anything right now because it's in a kind of a static state, so the sperm nucleus is very condensed and shut down and not much active transcription, if any, is occurring there. And then immediately behind these nucleus, two big round blobby things, those are mitochondria. And sperm have many mitochondria because they're going to need to generate motion ATP so those sperm can swim. Okay? So let's look at some events that are happening in the egg in response to the fusion of the sperm plasma membrane with the egg plasma membrane. So this represents the egg before fertilization, and we can see all these small vesicles hanging out here immediately under the plasma membrane. These are known as the cortical vesicles. Cortical for cortex, or outside portion of the egg. So these are marine organisms. They're in seawater, so the outside concentration of sodium ions is going to be very high, right? Sodium chloride for seawater. On the inside of the egg, there are a number of ions, both sodium and chloride, but in lower concentrations, but a high concentration of potassium. And so, in fact, there's a difference in charge across the membrane, the interior of the cell compared to the outside. If you actually put a volt reader on there and measured this difference, you would find that there's a difference of 70 millivolts difference in charge between the exterior and interior of the cell, with the interior being more negative. So when the sperm fuses with the egg cell membrane, this changes something. It makes the membrane more permeable to sodium. So when this membrane becomes more permeable to sodium, now sodium ions want to go into the interior to try to neutralize that negative charge, relatively speaking, that's into the interior. So sodium is going to rush into the egg cell. And this changes, so this difference in charge is known as the membrane potential. And once you have an influx of sodium like this, the membrane potential is going to change. So it's going to go from being negative 70 to a more positive value. And we can look at that on a graph here. So in this experiment, this is one of these done in a petri dish where you have some sea urchin eggs and you add in some sperm and you wait around, and pretty soon the sperm fertilizes successfully one of the eggs. And if you're looking at the membrane potential across that cell boundary, initially it was about negative 70, and then once the sperm fused, within a very, very quick time, just a couple of seconds, bam, that membrane potential changes and becomes more positive. That event somehow prevents any additional sperm from being able to fertilize or being able to fuse with the cell membrane. So we don't know exactly why that's true, but it does appear to be the case. So one of the things, another thing that a sea urchin embryo or egg has to be concerned with is that here we are in the open ocean, and there are tons of sperm around, but we only want one sperm to fertilize the egg. If two sperm fertilize it, that would be polyspermy instead of monospermy. So then you would have two sperm nuclei plus one egg nuclei, and that would be a triploid situation. And when that embryo tried to go through mitosis, it would probably run into a lot of trouble trying to evenly separate the same number of chromosomes into daughter cells and so forth. So for the survival of the embryo and species, we really need to have a situation, we need to have a mechanism for only one sperm to fertilize each egg. And the sea urchin does this in sort of a two-step process. The first part is called the fast block to polyspermy. And it represents what I just told you, where that we have a membrane potential change, and due to that very quick event, now no further sperm can fuse. Even though they're present, even though they're close, they might have already undergone the acrosome reaction, they're still not going to be able to finally fuse with the membrane. So they have a fast block to polyspermy. This happens within only a few seconds, but it's transient. So this will only provide a protection against polyspermy for a short time, say a few minutes. So the sea urchin also has a second, kind of slower but much more effective method to ensure that polyspermy does not occur. And that second method is called the slow block to polyspermy. Question?
What if another sperm touches it within the 3 seconds before the change?
So it could happen. Another sperm might touch it within 3 seconds or before something has prevented, such as the fast block has prevented another fusion. It is possible to get polyspermy, and especially you can see that in experimental conditions if the concentration of sperm are very high. So, these organisms are living in the ocean, and there's a typical kind of concentration of sperm that they would see and have evolved to live with. But if those conditions are changed, we can increase the incidence of polyspermy. And perhaps this just happens at a low rate in the natural populations anyway. So in that case, that embryo probably would not make it. Okay, so I have a fast block to polyspermy due to this change in membrane potential. So, what other events are happening here? So as the sperm fuses, another thing that we see is that calcium is going to increase inside the cytoplasm of that cell. And it actually turns out that instead of calcium coming in from the outside, the cell itself, the egg cell, has internally stored calcium in sequestered places, and now it's going to release it into the common cytoplasm of that egg cell. And as that happens, these change in ions are going to change the pH within the cell. It's going to go up. And all those cortical granules, all of those small vesicles bound by membrane that were right there on the very periphery of the egg, they are now all going to fuse with the egg cell plasma membrane. Just like the acrosomal vesicle fused with the sperm plasma membrane, now we have that same event happening on a very extended but smaller scale for all of the cortical vesicles. So they fuse with the plasma membrane and release their contents. So let's look at these events in a couple of slides here. So here we have on this slide red indicating calcium increasing inside the cell and blue indicating the pH increasing. And you can see that these events take somewhat longer. They take maybe a minute or two to happen, so they're slow. Okay? We can also look at these experimentally by using different sorts of dyes. So this is a dye that will recognize the concentration of calcium within the cytoplasm of the egg cell. And when that concentration is very high, the dye will be fluorescent and we can see it with our microscope. So in this series of pictures, this first one here is the egg cell before a sperm arrived, and overall the calcium level is very uniform and kind of low. And then once fertilization occurred, so that there on the left-hand side is where the sperm membrane and the egg membrane met together. And now there was a sudden burst or increase in the level of calcium within that portion of the cell. And then this whole thing spreads like a wave across the cell. So more and more calcium is being released from all the internal cells across the entire length or width or space of that particular egg cell. And as that happens, all of these cortical granules now go up and fuse with the membrane and release their contents exterior to this now fertilized egg cell. Okay. So here we had cortical granules, egg plasma membrane. Outside of that is the vitelline envelope, and outside of that is the egg jelly layer. So as this cortical granule contents gets released, it's going to be on the outside of the egg plasma membrane, okay? But between the egg plasma membrane and the vitelline envelope. So all of that contents here shown in orange is going to be now sandwiched in between the egg plasma membrane and the vitelline envelope. And in fact, there's so much of it that this actually makes kind of a new layer to the fertilized egg at this point. And moreover, the contents of this, which include a number of sugars and proteoglycans, they tend to want to absorb water. So some water is taken up. And effectively what this does is it creates a new layer known as the hyaline layer that's thick and obstructive and provides really a physical block that would prevent any additional sperm from being able to get through that so that it could get to the point where it could fuse with the egg membrane. So that's a physical block. Also, there are enzymes inside of this cortical granule that are going to digest proteins. And if they digest proteins on the vitelline layer, now there's no more receptors for that bindin to find and recognize and bind to. So the sperm are also not going to be able to attach to the vitelline layer very easily because they've lost their receptors, it got digested. Okay, so it's kind of a double protection here, and you can see why this might be very effective at preventing additional sperm from being able to fuse. Okay, any questions on this? Okay. So let's look here at this. All right, again, stymied by this double picture thing again. you a video, but I can't seem to get it up, so I will figure that out and show you after the break. Okay, so as the cortical granule contents are exocytosed, you're going to see in the embryo a visible space occurring between the egg plasma membrane and that overlying vitelline envelope. And because we love to name things in developmental biology, I just have to warn you. Now we're going to give the vitelline envelope a new name. So after it lifts away from the egg plasma membrane, we now call this the fertilization envelope instead of the vitelline envelope. Okay? And you'll be able to see this by an actual lifting away separation between those two layers due to the presence of all that contents of the cortical granules. Okay, so now we have a situation where egg and sperm are joined together. Each one of those cells contributed a nucleus, and so we have two haploid nuclei in cytoplasm that have to find each other, migrate together, and fuse. So these are called pronuclei because they're the precursors to the diploid nucleus. And there's a male pronuclei and a female pronuclei. And in a very short time, they seem to be able to draw together and fuse. And then this will restore now a diploid nucleus, and you'll be a cell that can go through mitoses and generate many diploid daughter cells, and that will be the beginning of cleavage, that'll be the beginning of a multicellular organism. I just finished that short. Okay, so this, at this point, is called the zygote, because we have our new individual has been created. So then sea urchin thus has two mechanisms to prevent polyspermy, the fast block and the slow block. And this is sort of a good study slide because it draws all this together into one summative picture here and shows you all the important events.